Okay, it is 9 o'clock, so let's get started with this seminar. We have an exciting program ahead of us. This is a mini seminar organized by the EU, by the Horizont Europe project BioEcoOcean, and the title of the seminar is Towards a Better Understanding of Marine Life. My name is Maria Koski. I'm a professor at the Technical University of Denmark, and I am the housekeeper of today, so to say. Just before we start, I have a few housekeeping things to say. If you want to present a question, please use the questions and answers function in the app. That is better than using the chat. The chat you can use if you have technical problems or if you want to chat with your colleagues. But please don't use the chat function for questions because they might disappear into the chat. And yes, so you find the question and answer function in the bottom of your screen, and just write your questions there. It would be helpful if you would also write to whom the question is particularly directed. And then we have these sessions after 1 or 2 talks where we take up the questions, and I will read them up then. And if, yes, thank you, Lina, you could share the screen. So our topic today is Towards a Better Understanding of Marine Life. We have an online mini-symposium, and if you put the next slide on, we can have a quick look at the program. So we have 4 slots here. In between, we will have the questions and answers, and each talk will be approximately 15 minutes. After 15 minutes, I'll start coughing really loud, or alternatively muting the microphone if that becomes necessary. I hope not. And yeah, and then after these sessions, we always take the questions and answers. And that is actually all what it is to it. If Lina, if you put one more slide on, then we can I can shortly present the speakers of the day. In this first slot, we have Lina, Florian, and Alexander talking about many exciting things. We have an introduction to BioEcoOcean. We have something about EOVs and fair data, and we have a talk about eDNA to detect the diversity of macroalgal forests. And next slide, do we still have? Oh, this is the first slot only. No, then we have the next slot where we talk about space-based monitoring of biological and ecosystem EOVs with André, and Sarah will tell us about pteropods in the changing ocean. And the next slide, please. Then we proceed to the biological carbon pump with Andy, and marine organic carbon atlas with Artur. And then finally, in our last slot, we have Lina back to talk about additional variables, and Anna to talk about connecting the dots, coordinating ocean observations. And with that, I would just like to wish you welcome and give the screen to Lina, who is our first speaker. Thank you.
Thank you very much, Maria. So, yes, I will tell you a little bit about the BioEcoOcean project. The title of this project is Co-Creating Transformative Pathways to Biological and Ecosystem Ocean Observations. And I'm Lina Mtwana Nordlund. I work at Uppsala University in Sweden, and I'm also an expert member of the Biology and Ecosystem panel at the Global Ocean Observing System. So BioEcoOcean is a 4-year project funded by the European Union. We are a quite small consortium, but powerful. And the ultimate goal with BioEcoOcean is to enhance the biology and ecosystem ocean observing capacity for advancing scientific understanding of the ocean and increase the utility of ocean observations. We believe that we need coordination and interoperability, but the big question is, how do we do it? And we think that one of the solutions is more consistent communication and collaboration among sectors and stakeholders. So therefore, we are co-creating something that we call a blueprint. A blueprint, and this blueprint is going to be a question-based support tool. It's a co-creative process, so it is under development. And this question-based support tool to help us think a bit more holistically about the ocean and how to understand the ocean and how to monitor the ocean. And the reason why it's question-based is because we really wanted to spark and encourage collaboration. It doesn't tell you the answers, it gives you the questions that you might need to ask yourself, ask your partners, or maybe contact others to help being able to solve these questions in a context that is suitable for you. The blueprint also aims to be a support for communication across sectors, and will also identify where collaboration would be really advantageous. And it can also be used as a sort of future vision tool for more integrated approaches and for planning. So the blueprint, it will be launched on a digital platform. It will be interactive with downloadable resources and also with a lot of learning resources that can help you in observing the ocean. The launch is planned for September 2027, and it will be hosted under the UNESCO umbrella. And as I mentioned, this is a co-creative process, so it's evolving. We started just over 1.5 years ago, moving from a more of a linear process that was already quite advanced compared to what was out there already. And now we are at this sort of spider web, trying to illustrate that everything is connected. We're not really sure yet where it is going to take us, because we do have a lot of co-creation left to do, but it's really, really exciting. And the reason why we are using this truly co-creative approach is when you start talking to people, it's a very powerful way to identifying and realizing challenges sort of in your workflow when you're observing the ocean, and also finding these solutions and these new pathways that might need to be developed to do this more effectively. And the blueprint has a lot of different components, and these components really try to bring out a lot of different aspects from what is needed to do truly successful ocean observations. Oh, I'm sorry, there's supposed to be a red box around data collection and data management there, because I want to just illustrate that you can have this direct connection maybe between the data and then where to maybe where to publish this data. Or as a data publisher, you might not be able to host the different types of data types like pictures or videos or interview data that a person wants to deposit. So we need a strong collaboration and discussion and development here to ensure that we have better data management, so our precious collected data can be useful for many others. Another example is, for example, the connections between data collection and the data products. Are we actually collecting the right data for the data products that we need in society, that policymakers do need? So there's a lot of connections, and there's a lot of connections that we might not think about. And hopefully these questions can help us and support us in thinking a little bit more holistically about ocean observations. So there's a lot of different questions. I want to bring up one here, for example, in the planning phase. Something I think that we might often forget is like, who else is doing this kind of data collection or similar data collection? When and where are they doing it? can we collaborate somehow to sort of maximize the output? And there's multiple ways to be part of this co-creative process. If you visit our website, you can either scan the code or someone will share it in the chat here. And then you can sign up either for our newsletter or joining upcoming online blueprint workshops. And I want to move a little bit from the blueprint to EOVs. So EOVs are essential ocean variables. And I think we're all very painfully aware that we can't measure everything at all time because it's too expensive, it's too time consuming. So this essential variables concept is sort of born from that there are some variables that might be very important to measure, and it can give us quite a big understanding. So we have the EBVs, which are essential biodiversity variables, ECVs, the essential climate variables, and EOVs, the essential ocean variables. Sort of that is really one of the foundations in this project. But just as an example of these essential variables, I think we're all aware of the weather forecast. And I think this is what we see here on the screen is something that is very nice, and it's like an application in society, right? We can just go in and check the weather. When is the sunrise? When does the sun go down? But just thinking about having quite accurate weather reports, there is so much behind producing something like this. First of all, we need to measure temperature in the same way. We need to make the data available. It needs to be gathered somewhere, and it needs to be analyzed. We need to make forecasts, etc. And I mean, the foundation here is really that we're actually measuring the same thing in the same way. So this is sort of the idea behind the essential ocean variables, or all the essential variables, is that we measure the things in the same way. We share the data, we make it available, so we can contribute to a larger scale. And these are especially then for sort of the global level. These essential ocean variables are governed by the Global Ocean Observing System, which is part of IOC UNESCO. And in BioEcoOcean, we are really working hard on trying to accelerate and improve the implementation and usage of these essential ocean variables, and especially for biology and ecosystems. Because if we are starting to measuring things the same way, and we have comparable data and available data on a global scale, we can really take a step forward understanding our oceans. Each of the essential ocean variable has a specification sheet. It's basically a document pointing out which variables are recommended to be measured. And this is we have in BioEcoOcean, we have supported this process of updating all these 12 EOV specification sheets for the biology and ecosystems. But the information in there is based on the global community, and it's a lot of people from around the world that has been part of sort of defining what should be measured and how it should be measured. It also has a very strong data section on how to actually manage your data so it becomes available and can be used then in global assessments, for example. So these specification sheets, they they have a lot of these different components and also sort of the justification and also which larger global frameworks it can contribute to. So the key here, I would say, is really that we do a lot of observations around the world, but we need to publish that data so it becomes available so we can make really interesting data products. So it's actually quite important where we publish our data and how we do it and what we're actually publishing so it can be used by others and also that we can get credit for the data that we are collecting. And part of BioEcoOcean is also the Ocean Biodiversity Information System. So we are developing a lot of sort of resources to help the global community to make data more available and publish data. But now I want to pass over to Florian, and he will provide an example from the Baltic Sea. So thank you very much.
Thank you, Nina. Lina. And now then we just go on with Florian. And please remember to write your questions in the questions and answers box.
All right, I hope you can hear and see me. Thank you for having me. I am Florian Lüskow, also from Uppsala University, and my talk will directly feed in what Lina just mentioned. So how we can actually use local observations to contribute to global assessments, especially using the biology and ecosystem EOVs through FAIR data. And as Lina mentioned already, I will start with an example from the central Baltic Sea. That's where we are located on the Swedish island of Gotland, where we did a survey to estimate the abundance of jellyfish. Surely this has been done in other places, but we were the first doing this in this part of the Baltic Sea. So our local goal was to publish those data, but there's more to it. If the data are published in a fair manner, so they are findable, accessible, interoperable, and reusable, they can contribute to a much bigger goal, and we can achieve so much more than what we just wanted to do locally and regionally. And for doing so, it comes in very handy to use these so-called essential ocean variable specification sheets. Lina touched upon them already, and I don't want to repeat much of it, just saying that they have a lot of information regarding the background of the target group in the case of the jellyfish, as they are part of the zooplankton. We look into the zooplankton biomass and diversity specification sheet, but they also put it in a perspective of larger frameworks, such as the Sustainable Development Goals. But to help you to identify what you actually need to measure to produce such comparable data, they have a set of subvariables that should be measured. For instance, the biomass, the abundance of a specific taxon, and the species composition of a zooplankton assemblage or community. And it also gives you an idea about supporting variables, so other measures that are useful but not necessary for telling the story, such as sea surface temperatures, surface salinity, oxygen concentrations, and others. It also gives you a perspective how your data can be used in larger contexts, such as analyzing the community and the population status and potentially trends. This can be done on a regional or a global level, but also about biogeographical shifts of populations or whole species, again on a regional and a global scale. But what does it really mean for your specific data? Because this link from local data to contributing to global assessment is still a little bit difficult, we came up very recently with a step-by-step pipeline for publishing such fair data. And again, this is done for our case in the Central Baltic Sea, the jellyfish. So where it gives you guidelines for what to consider and how to structure basically everything you do, starting with the data collection and the way how you record metadata, the date, the time of the year, the GPS locations, the collection methods, but also when it comes to the data formatting. Because a field notes sheet is very different to what is actually required for publishing fair data. It follows very specific standards. The Darwin Core is one of them, which requires a so-called controlled vocabulary and uses specific identifiers to really make it interconnected and interoperable with other data sets and with other users. And in this database, the Ocean Biodiversity Information System, you're not alone. There's already a lot of guidelines available in published form, but also experts available to support your endeavors. In step 3, in publishing these fair data, you really want to make sure that it's easy for any user globally to find your data and to access them. Ideally, you want to get credit for it, so they should have a DOI, and you want to be sure that they are published in a very comprehensive matter. So they are complete. What do I mean with this? The data are published alongside with the metadata. And eventually in step 4, the use and access of these data, you want to have it easy to say, okay, I'm interested in a certain time period or on a spatial scale. And a polygon could be a data, a specific point, and all kind of data could be included in this. Could be biomass and abundance in the case of the jellyfish, but it could be also acoustic data, imaging data, or tracking data. obis.org basically is a one-stop shop for all of this. And there is R packages already out there, so you don't need to start from scratch. And again, there's a support staff that is very happy to help you with all of this. And if you want to know more about this particular pipeline, you can find more about it in our recently published paper, which is also accessible with the QR code. And quickly going to OBIS, the website, it's very easy to find those data. So in the top right corner, you see buttons like data, resources, or the search function. And if you would go for the search function specifically and would enter Gotland and jellyfish, you would be directed towards this data set, which is then one-to-one linked to our paper that I just showed. And here you have a very nice overview of the data points that is out there. 60 abundance estimates were connected with, 510 metadata records. And well, there's a description of the data set and the metadata available. But bring this back to the global scale. This is just one data point, or 60 to be correct, but it's very regionally confined. So of course you want to connect it to something larger. And one way of thinking about this is, well, if everybody across the globe would do this, publishing the data, if it's a specific point in time and space, or even long-term time series, we can be very powerful, because then we can start creating a larger picture, which, for instance, they did in this 2012 paper to assess whether there is global trends and changes in the jellyfish population. Each circle represents one time series, so one data collection event or series with different lengths, which is indicated by the circle size. And each of them, of course, has a power in itself. But putting all of this together, we really can start understanding, do we see an increase in jellyfish populations globally, as indicated with the warm colors, or do we see a neutral trend, or maybe even some decrease in populations indicated by green and blue? So the power really is to make the data available and fair, and then we can take it from there. Thank you.
Thank you very much, Florian. And then let's go straight to the last talk of this slot, that is by Alexander McGrath. And Alexander, please, the screen is yours.
Awesome, thank you so much. Good morning, everyone. My name's Alex. I am a postdoc at the University of Pisa. And today I'm going to be talking to you about some of our work about using eDNA to detect the diversity of really important macroalgal forests. But before I dive into our work, I want to set the stage with a broader look about how environmental DNA, or eDNA for short, is transforming how we study marine life. The ocean is the largest living space on the Earth and is home to an extraordinary biodiversity of organisms and animals, which supports fisheries, coastal protection, nutrient cycling, and global carbon storage. But marine ecosystems, globally, are under pressure from climate change, warming waters, invasive species, and habitat loss. In order to protect these systems, we first actually have to understand what's there. And to do that, we need a reliable, efficient way of measuring biodiversity. And traditionally, this has been actually a major challenge for a lot of our efforts. So, marine ecologists have relied for decades on diving surveys using quadrats and transects effectively to go underwater and directly measure what's there. But these approaches can, whilst being extremely valuable, are often very expensive and potentially quite dangerous for those participants being involved. They're labor intensive. They also require expert species identification skills, and they often miss cryptic, rare, or, I mean, if you're diving during the day, which traditionally most diving is done, you're missing a lot of nocturnal species as well. Furthermore, as you can only be in the water for a certain period of time, you can't always see how species abundance or composition changes over time unless you're there every day, which is very labor intensive and very expensive. These limitations have pushed ecologists and biologists worldwide to develop new tools, especially molecular ones, such as environmental DNA. So fundamentally, what is environmental DNA? Well, organisms living in the ocean, much like us on land, shed scales, feces, gametes, and they float off into the water column. Environmental DNA at its core, at its most simplest point, is purely sampling these things, is going and seeing what's floating around in the water. This is typically done either from water or the sediment, and which is then sequenced. The DNA is then sequenced to see which species are present. So if we go through the pipeline, we have our beautiful little fish here. It loses a couple of scales as it's swimming through the water. They float into the water. We then come along, fun scientists, and we take a volume of water, which depends on your species of interest, which we then push through very, very small filters that not only capture larger material like scales, but are small enough to actually capture components of raw DNA as well. These are then extracted and sent for sequencing, which effectively gives us a fingerprint of that organism that we were interested in the start. Yeah. So one of the really big positives and exciting things about DNA, eDNA, I mean, DNA is exciting in itself, but the exciting things about marine eDNA is its applicability across different ecosystems. It's a basic approach, which is very easy to carry out. You just need a bottle, effectively. You can go with a 1 liter bottle of water and take a water sample. And as long as you have the lab equipment or send it to someone who has the lab equipment, you can get an assessment of what's there. So in coastal and estuarine systems, eDNA can be used, for example, to monitor fish communities for fisheries management and to detect invasive species that can potentially threaten their systems, long before traditional surveys are actually able to detect them when they're actually visible. Within the pelagic open ocean, it's very difficult to get a proper assessment of fish stock due to the fact that it's a very large space and typically fish aren't everywhere all at once. And during oceanographic surveys, eDNA can be taken alongside, as I mentioned, just by a sample of water or sediment, which can help map biodiversity over enormous areas and detect migratory species such as marine mammals or sharks. Within corals or coral reefs, you think of all the beautiful complexity that's here within a reef. There's things living with all these nooks and crannies. It's very, very difficult to actually see them for one, and furthermore identify them down. So eDNA can be really useful here to actually assess cryptic and rare species. And probably one of my favorite uses of eDNA is actually within the deep sea. I'm a coastal ecologist, I'm an intertidal ecologist, so I don't do anything with the deep sea, but it does still really excite me. Because traditionally, when we think about the deep sea, sampling is incredibly difficult. You're at an extreme depth with extreme pressure. And it's really, really difficult to sample with normal tools. However, with eDNA, sorry, I'm sounding like I'm trying to sell you all on eDNA, but eDNA can help us identify species that we can't see. Like, you take a sediment sample and you get the diversity of all of the beautiful polychaetes that are living there. But importantly, for today's talk, I'm going to be talking about my favorite ecosystem, kelp forests or macroalgal forests, wherein eDNA can be used to assess the incredible biodiversity associated within them, and which is especially important for foundation species whose survival and persistence within, oh, no, I went too far. Survival and persistence within a changing ocean underpin the survival of all the beautiful critters that live within them. So macroalgal forests are these beautiful marine ecosystems formed by macroalgae, so large algae, and they effectively form a similar role underwater as to what we normally attribute terrestrial forests. So they form nursery habitats for fisheries. They sequester carbon at an incredible rate. And they protect coastlines from erosion and also wave impacts. But as I'm sure you can imagine, they're actually very, very difficult to monitor. They're typically very dense structures, which makes it difficult for divers to actually see all that's there. And also a lot of the critters, especially in the Mediterranean, we're working with macroalgae that is 30 centimeters high. And a lot of on a good day, 30 centimeters. And we're working with critters and animals which are a couple of millimeters or a couple of centimeters across. So actually seeing what's growing within a macroalgal canopy can be very, very challenging. That's where eDNA really comes in and can be a very, very powerful tool. Because it can detect species that are hard to observe, such as our beautiful little nudibranchs or hermit crabs, present only briefly, like some of the rare fish taxa that we get coming through, or living within complex three-dimensional spaces like these this beautiful sargassum. So, diving straight into our research, we're based in Italy, in Tuscany and Pisa. But the majority of our work is actually conducted on this lovely archipelago of islands, the Tuscan archipelago. And the work I'm going to present today was actually, sorry, I'm jumping ahead, but something didn't pop up. So our work was conducted primarily on 3 islands, so Capraia, Pianosa, and Janutri. This is just some images of our beautiful ecosystems. This is Pianosa on the right. Is it Pianosa? I think. Or Janutri, anyway. So our work is conducted across the Tuscan archipelago, where we've done a series of eDNA sampling campaigns to try and test a couple of questions. Firstly, how effective is eDNA as a sampling tool versus traditional methods? And secondly, what is the detection range for sessile organisms? So for algae, how close do you have to be to that algae to actually detect that it's there? Oh, I've got a duplicate. So our 3 islands, forgive me. So for this first piece, wherein we were assessing traditional versus eDNA sampling, we had 3 islands: Capraia, Pianosa, and Janutri. And within each island, we had 2 sites, typically north and south, but that's 2 spatially discrete sites effectively. And at each site, we had 2 depths, because we also not only wanted to assess whether we could detect it right next to the algae, but we also wanted to see whether, say, for a manager coming along, whether they could just take a bottle of water at the surface and they would still get the same signal. So our 2 depths were surface and then at the bottom. And then within each of the sites, we have 2 areas per site, which are separated from each other. And it's relatively shallow, so only about 7 meters. And so within each of the depth area combinations, we had 3 replicates wherein we took 1 liter of water, 2 photographs, and an algal sample. And I haven't touched upon these algal samples yet, but they're really important because one of the main drawbacks of the eDNA is the fact that you need effectively a reference database. You need to say, this species has this sequence. But for a lot of our Cistoceta species, so our species of interest within the Tuscan archipelago, we don't actually have a very good library in the back of which to test against. So what we might be seeing is when we go and collect water and we sequence, we might actually have Cistoceta there, but because we don't have this library in the background, we can't actually say for sure. For the second goal that we had, which was testing distance, this is just a little schematic showing that. We sampled at the bottom within the canopy. It was 10 meters rather than 25 meters, and then 50 meters from the canopy, effectively trying to assess, as I mentioned, at what distance we can detect macroalgae. So we saw some really, really nice results, some really promising results, actually. We saw that when comparing eDNA with traditional photograph sampling, we saw that on average, we had significantly more species of Cistoceta, so our habitat-forming macroalgae, than we were able to detect with our photos. This is another thing with Cistoceta. Sorry, I should have mentioned it earlier. It's a very difficult species or a species group to detect from photos. It's a very, very morphologically complex group of algae. But this is a really nice result in the fact that we have more detections of species within eDNA than in photos. And then for that that secondary goal of seeing whether we could detect the macroalgae at the surface rather than having to put divers in the water, again, we had a really nice result. I was really happy with this, really excited, because as you can see, they're pretty analogous to each other. The number of species detected at the surface versus within the canopy. This is for eDNA. Sorry. And we saw a really nice relationship forming. We saw that when we have increased eDNA abundances, we actually have an increased cover. This is for Cistoceta funiculata. So it's actually relatively easy to identify from photos compared with the others. We saw a nice trend forming, so we can actually be quite confident and quite sure with our results of, if we actually have more eDNA, there is more cover within the area, which is an exciting thing. So to conclude, eDNA, oh, sorry, I should mention before, that second goal of distance from canopy, we only did the sampling this summer. So the little DNA is actually currently being sequenced. So it's off out of our hands. So hopefully in the next couple of years, we'll be able to share with you the effective distance on DNA dispersion. So in conclusion, eDNA has provided a relatively low effort but highly accurate tool for biodiversity measurement within our system. And I should also stress that the work we've done is focused on the canopy algae, but within our sequencing reads, we have a huge amount of diversity of other different algae. So all of the small epiphytes and also some of the algae growing beneath the canopy, which is promising. However, we need a lot more sequencing work getting that back-end library for macroalgal species, especially within the Mediterranean. Other areas of the world with more charismatic macroalgae have far better reference libraries, but we need to put in a lot more work sequencing macroalgal species. And also, touching on this, we need to develop a tool that determines, that actually links the number of sequences we get. So say I get 10 sequences of one algae, what does that actually mean in algal cover or in the term of the ecosystem? Yeah, so with that, I want to thank Alessandro and Luca and all of our lovely divers that have helped with this work. And I think we're jumping onto questions now.
All right, thank you very much, Alexander. We have a good time, 8 minutes precisely for questions, and I can see that there are some in the questions and answers box. So if you just start from the top, Lina Ebarkina is asking mainly to Alexander, but I'm sure Lina is also willing to comment on this. Can you please share your perspective on the environmental impacts of data collection and how to address them? Maybe if Alexander, you could answer first, and then Lina and Florian could comment further.
Thank you for the question. And it's a really important thing we have to think about as ecologists. We're wanting to protect these systems, but what is our impact in actually saying going and ripping out algae? And I think that is another really strong positive for eDNA is the fact that it's non-invasive. You go and you take a water sample and you get your data rather than having to go and collect the physical algal specimens to identify them. Although there is the caveat as I was mentioning with the library preparation. But rather than having to go and collect large amounts of algae, you can do it with eDNA. So it's just water for us at least.
Thank you. Maybe Lina, if you would like to add something to this.
Yeah, I think this also really makes the point that if we publish the data, I mean, every time we go out, we do some sampling, there's some kind of impact on our environment. So sharing the data, making sure that others can also use this data, so we sort of maximize the impact of every single sample that we do, I think is extremely important. And we really need to shift the way we think about sharing and publishing data. I think everyone that shares data should really be celebrated because the raw data is the gold, right? It's the most important thing because based on that raw data, you can make a lot of really cool products and use for purposes that you might not even thought of. Thank you.
Thank you. Florian, do you have something to add?
Yeah, very quickly from me. So no one method is perfect. And so combining non-invasive methods like the eDNA, which has weaknesses, with other methods like acoustics or visual methods is actually the key to get a holistic view. And so combining in all kind of setups, different methods, I guess, is the key to the success.
Thank you very much. Then we have a number of eDNA specific questions to you, Alexander. I will read them aloud here. We have Martina Gagliotti asking, did you investigate only algae species in the eDNA samples or also animals?
So for the purpose of this work, we were really focused on the algae. And in order to, I mean, we can look through our sequences and there are, when we initially made the library to see what species were there. There are like crustaceans and stuff that we can pick up. If we want to pick up more transient species like fish, and some of the larger fish especially, we need to collect more water, which is one of the drawbacks of what we've done. We've only collected a small amount of water. I mean, it's fit for our purpose, looking at algae, but if we want to collect or detect more transient species, we need more water.
Thank you. Then a little more on the lines of eDNA. So the next question, what is the limitation in terms of time and detection limit? I guess time would mean the collection time, but also in the next question there, the time, how long will the eDNA stay in the water? So the question goes, if I collect an eDNA sample from one location where the organism fish has already left an hour ago, can eDNA still detect its presence? So the limitations in terms of distance and the time.
Really good question that gets at two of the main gaps within our knowledge about eDNA at the moment. I can only really speak to our algal work, but and also, as I mentioned, we're still waiting for sequencing back for the distance element of that. But from the amount of DNA that we were getting on our filters, it was pretty equal across from canopy out to 50 meters. I know there's been some work, there's a whole host of work being done in the northern Mediterranean up near France, where they've been able to detect very rare shark species on a very, very broad scale. Whether you can detect a fish that's been there an hour ago, I think it depends on whether the fish has dropped a scale or has created feces or something. I think that's why it's really important for eDNA, for our type of sampling, is if you're wanting to actually get detections of these species, there needs to be consistent temporal sampling, like in situ long-term sampling. And there are passive detectors, so you effectively put a box in the water and it detects at set time intervals over however long you want, effectively. Yeah.
All right, thank you very much, Alexander. There is two more questions, very specific ones, and I wanted to ask if you, because of the time, could please type the answers into the question and answer box. And then just before we close the first slot, Lina and Florian, I was wondering, you also do work on the eDNA a little bit. Do you have would you like to comment on these questions about the time and the distance as well?
Yeah, when it comes to fish, a lot of the literature says that the DNA can stay, eDNA can stay up to about 24 hours in the water column. But I think there's a lot more research that needs to be done on this, absolutely. We've also been looking at sort of the top-bottom, comparing the top-bottom when it comes to fish, and we do see some differences. So we are really recommending sort of sampling the whole water column, and hopefully there will be a paper coming out about that quite soon.
All right, thank you very much. Then Alexander, if I could ask you then just to type the questions into the question and answer box for the remaining ones. And then we proceed to our next slot. And we start with André Valente. If you could please share your slides. And in this slot, we have again two speakers. We have André Valente and Sarah Albernhe. And yeah, I think we can just get going. So André, please.
Sure. Thank you. So good morning to everyone, or good afternoon, or good night to some of you. My name is André Valente. I'm going to present today on this topic of space-based monitoring of biological and ecosystem EOVs. So I'm from the Atlantic International Research Center. We have headquarters in Terceira, but we are an international organization with members across the Atlantic. We work on several areas, and today I'm going to give a very broad approach on this topic, presenting some of results that we work on and some other works from other researchers. So as Lina already mentioned, the essential ocean variables are basically variables that have impact. So and they can be feasibly to measure. So it's possibly technically and economically measurable. And today I'm going to focus on these biology and ecosystem variables, which are also the focus of the BioEcoOcean project. And I'm trying to minimize here my window. Okay. And I put here some arrows because almost most of these variables can be, we can derive some information from satellites on them. But some of them, for example, fish, sea turtles, seabirds, I put there in red. It's more difficult to I'm going to say direct and indirectly. Of course, you don't measure this directly. It's always through light or some other source of energy. But these fish and sea turtles and seabirds, you cannot see them from space, but you can access variations on them, for example, using other satellite-derived products such as sea surface temperature, sea surface currents. So you can understand their distributions through these indirect products. But the others, you can try to access them. And today I'm going to, since I only have 15 minutes, I'm going to focus on 4 of them. So I'm going from the smallest to the largest, from phytoplankton to zooplankton, macroalgae, and then marine mammals. So it's first important also to explain that when it comes to sensors on satellites, you have 2 types. So the passive ones, which they measure energy that is either it's emitted from the sun and reflected on the Earth, such as the visible part, and then also the energy that the Earth emits. For example, these infrared and microwave radiometers, where you get sea surface temperature. And then you have the active instruments that they launch their own energy. So the altimeters, the radars, the spectrometers, and also the LIDAR. In terms of biological observations, most of what is done is based on the optical and visible radiometry. I'm going to touch upon a little bit on the LIDAR for zooplankton, but the most of it's from optical visible imagery. And in terms of these sensors, historically, they can be divided in multispectral and hyperspectral. Multispectral, they measure the radiance in discrete wavelengths that are relatively wide. And historically, the low resolution has been what has been mostly used because it was open source. These usually have 1 kilometer resolutions and large swaths, so you can observe very large areas of the ocean almost every day. And then recently, we started to have high resolution, also open source. For example, Landsat with 30 meters has became open source around 10 years ago, more or less. And Sentinel-2 also more or less 7 years ago, it was launched and also made open source, 10 meters. Then you have the very high resolution. So these are the commercial. They all have always existed. Now we have until, I think the maximum is around 15, 30 centimeters that you can buy from world view. Then you start to have hyperspectral. For example, now PACE, launched 2 years ago, you gives again 1 kilometer hyperspectral resolution, which is very good. Then you start also to have high resolution hyperspectral, so around 30 meters pixel resolution. These, of course, have a smaller swath, so you see less, and also more separated in space. And this is data that is coming and can be will be used more and more. So in terms of EOVs, phytoplankton biomass and diversity is the main, let's say, EOV that space has been addressing, and it's more mature. This is because one of the key EOVs derived from space is chlorophyll concentration, which is a traditional proxy for phytoplankton biomass. And this has been operationally from NASA since 1997, for example, and now Copernicus also has joined this, producing chlorophyll products regularly, every day. So all the community uses much of this data for different modeling and other applications. And this is also expected to continue with new sensors. And this also, the chlorophyll from satellite also is an example of the collaboration and standardization that you want for a biological EOV. For example, there is this compilation where, well, myself, with many of the community, we compiled all the data that was freely already available in many repositories of chlorophyll and also other data sets. And it was published in a, let's say, a simple table with each row being one unique station. And this can really help advance the field and serves also an example of what we can do for the other EOVs. Of course, EOV phytoplankton biomass with satellites is much more than chlorophyll. There is many works on community composition, which is and blooms of some species, and this is now expected to increase with this new hyperspectral mission PACE, namely, that is still in an experimental phase, the products, but I believe they are already being used by many of the researchers. Now I'm going to pass to zooplankton. So this is a less explored area, let's say, but also because of the difficulty, because there has been some works that I put here, some examples, because they take advantage that, for example, Calanus finmarchus, when it gets really reddish and in a stage where it grows a lot, you can have these swarms that even fishermen, there is stories that the water gets red coloration. So there is an opportunity here to also observe from satellite. Also LIDAR, which is a technology that it's not really designed for ocean applications. There has been some studies that looking at, for example, day-night difference in the backscattering coefficient, looking for diel vertical migration signals. And for example, in our center, we are also here involved in one project, Clean Blue Feed, where you had a autonomous vehicle with the echogram in Origin C at the time of the bloom, with the echogram signals, looking for the zooplankton. We are now trying to correlate this with ocean color PACE and also LIDAR, but from ICET2. So these are photon heights that you can derive some information from below the surface. And now I'm going already passing to macroalgae cover. So macroalgae canopy cover, possibly the more mature is the surface canopy formic macroalgae, such as the giant kelp. There is already this global application of Kelp Watch. And it has take advantage of these high resolution Landsat and Sentinel-2 sensors that you can detect at a wide scale the coverage of this emergent part of the kelp. At the intertidal area, you can also explore. So this at sometimes in intertidal areas, the forests, the kelp forests are exposed. In this case, in BioEcoOcean, we are focusing on the Atlantic Living Lab, which is in the northwest Portugal. There is some extensive beds of macroalgae at low tide. And we are developing different methodologies, taking advantage of also historical flights and also flights that we are doing in BioEcoOcean with drones. So we are merging the drone very high resolution with the Sentinel-2 low resolution. So we build models based on the UAV data. And then we apply this over large scales that the drone cannot access. So the drone is very specific in a very small area that you see in the right. And then we expand this area with the satellites and also over time. Of course, there is also the subtidal macroalgae that it's always submerged and it doesn't get exposed. And there are also many research on this, and there has been a let's say a focus as seagrass from the community of remote sensing for a long time. And it's usually with very high resolution imagery, just because it's challenging. The light gets absorbed very easily. So you need to focus on shallow areas. You need to understand well where the patches of these macroalgae are. Usually a standard procedure is you build polygons of your constituents of the benthic constituents, and it's important that you classify or you understand all of them, so that your classification is more let's say with more confidence. And while it's not let's say specifically addressed with the EOV, you have this floating sargassum, which in the left side there is also quite research on this, and already several tools that map this floating sargassum across the Atlantic. And also you can also look at the beach cast. So when the either sargassum or also some other species that are attached to the bottom and get released at some times of the season. For example, you see in the right, this is an invasive species that progressed from the Mediterranean and reached Azores in 2019, and it's already occupied much of the bottom. So the regional government is worried with this. You can see here very large accumulations of biomass on the beaches from these invasive species, and you can detect this with satellites and build information on this. Finally, marine mammals. So it's possible. Usually always with very high resolution imagery. Ideally, the highest, the best, 30 centimeters. You can do this in several ways as a visual method. You scan all the image, or with an automatic way. For example, we have a project in New Space where Portugal are launching a constellation of satellites at national level, and we are testing if 1 meter resolution was sufficient. And so the basic automatic way is you develop a training data set, and then you apply these deep learning models. And then you divide the image in several tiles, you classify each tile, but then at the end, it's always important to have a supervised classification. So as an expert, you scan all the positive detections and you filter out the ones that you consider that based on your experience that are more likely. And here I showed 2 examples that we got from blue whales. And in fact, we had also contacts from whale watching, which also supported our analysis. And just to finish, I'm going to say that the hyperspectral sensors are really going to change our approach in many fields. So there are several planned missions from NASA, also from Copernicus. So there is good opportunities here for improving our knowledge, and also in general we can expect more data in general and also better quality. For example, Sentinel-2 next generation could be even less than 10 meters. We don't know. If so, it's a good advantage to have this as open source, because you can just monitor over time easily with open source data. But at the same time, will be more very high resolution data from private companies and national constellations. So this can also basically, in a way, change the landscape of how we have access to this data, which it's also, again, opens opportunities to better map several of these EOVs. Finally, you will have new tools. For example, imagine a ChatGPT where you ask to map in this case, it's a land application, but you can maybe imagine that you can ask Google to map your seagrass, your macroalgae, and it can give you a really starting point to your work, and you also have much more organized data in terms of data cubes and digital twins. So that's it. Thank you very much.
Thank you very much, André. I suggest we just jump directly into the presentation of Sarah. If you would share your screen, please.
Yeah.
And please remember there is open the questions and answers box, so just type your questions in there and we take it all after Sarah's presentation.
Can you see my slides and can you hear me?
Yes.
Good. Good. Thank you. Good morning, everyone. My name is Sarah Albernhe. I'm a postdoctoral researcher at Mercator Ocean International. And today I'm going to talk about Sentinels in a changing ocean, modeling pteropods as early warning signals of climate change. So as you may all know, human activities have caused global warming since the pre-industrial period, leading to rising temperatures and diverse environmental changes. For instance, the CO2 uptake by the ocean has significantly changed the ocean chemistry, leading to the acidification of the ocean. And this acidification has strong impacts on marine organisms, creating winners and losers. So for instance, a group of organisms that is particularly vulnerable to ocean acidification is the marine calcifiers. So for instance, we have the corals. They are organisms that are able to produce calcium carbonate to form hard structures like shells, skeletons, or exoskeletons. And among these organisms, we have the pteropods that we're going to talk about today. So who are these pteropods? On the right-hand side, you have 4 photos of different species of pteropods. They are small mollusks of a few millimeters, and they are part of the zooplankton. So if we consider a simplified food chain in 4 levels in the ocean, the first level is the phytoplankton. So it's the plant-like plankton, it's macroalgae, and they are eaten by zooplankton, so the animal-like plankton, which is the prey of the micronecton. It's a group of marine organisms from 2 to 20 centimeters, which is the prey of the top marine predators like tuna, sharks, marine mammals, etc. And so our pteropods today are part of the zooplankton component, which is a key component of marine food web, as it's located at the intermediate level of the chain, and so it sustains all of the higher trophic levels. So you may have noticed on the past slides, some pteropods are wearing shells. They look like snails, and some are not. So today we're going to focus on the shelled pteropods. And so they are able to produce their own shells of calcium carbonate. They are filter feeders, so they produce their own mucus net around them that act as a trap for particles. So it's going to trap, for instance, phytoplankton, and the pteropods are going to eat on these trapped particles. To provide them locomotion, they have a pair of wing-shaped lateral appendages, as you can see on the photo, that have earned them the nickname of sea butterflies. And these organisms are drifting with the currents, so they are not able to swim against the currents. But they are having a very interesting behavior, which is called diel vertical migrations. They are able to migrate every day between the surface and the deep ocean. So they stay during the day at depths to hide from predators because there is no light, so they avoid visual predation. And they go back at the surface during the night to feed. And they perform these migrations every day. So here you have an example of the California currents. It's modeled pteropods. And you can see on the left-hand side that during the day, they remain at depths. And during the night, they go back at the surface. And so performing these migrations, they are exporting and sequestrating carbon in the deep ocean. So these organisms are very small, but very important. Now let's see why we call them sentinel species. The ocean acidification is increasing the dissolution of the shells of the pteropods, which is reducing their survival. So here you have an example of 4 photos derived from laboratory experiments. And so it's a time sequence of shell dissolution in the water conditions, so the acidity conditions that are projected to happen for the Southern Ocean by 2100. And we can observe that in 45 days, we have a severe shell dissolution. So it's very important and very fast. And knowing that, the habitat, the suitable habitat of the pteropods in the ocean is driven by 2 main factors, the temperature of the water and the acidity. So the pteropods have to live in waters where the temperature is good for them and where the acidity is not too strong, not to dissolve the shells. So here you have a figure of the suitable habitat for shell pteropods as a percentage of global ocean volume from 0 to 300 meters. So basically, what does this figure mean? It means that in 1998, the pteropods could inhabit in nearly 35% of the ocean volume. And this surface, this habitat has decreased since then. And it has decreased by a surface that is equivalent to 70% of the Mediterranean Sea, which is a lot. So that's why we say that pteropods are sentinel species. They quantify the effects of global warming and ocean acidification on marine ecosystems, on marine organisms, sorry. We also know that the acidification threat for pteropods is stronger in high latitudes. So here on the map, you can see the green dots that are areas with no evidence of shell dissolution. And the red ones are areas with severe shell dissolution. And the red dots are located in higher latitudes. So for BioEcoOcean, we focused on the oceanographic station Papa that is located in higher latitudes. So if we have a look at the environmental conditions there, in orange, you have a time series of the surface layer temperature at Papa Station between 1998 and 2020. And in blue, you have a time series of the aragonite saturation, which is a proxy of the acidity. The higher the aragonite saturation, the lower the acidity. Okay. And you can see a gray slice on the time series that correspond to an interesting event called the blob. It's a marine heat wave that happened between 2014 and 2016. And you can observe a dual change both in temperature and acidity levels, but with antagonist effects. So we can observe a temperature rise and an aragonite saturation rise, which means lower acidity. And so we are wondering, how did the blob impact the shelled pteropods? So we also have a time series of observations of pteropods, so from zooplankton nets. So first we can observe quite an important interannual variability with very high peaks. And during the blob particularly, we observe that we have a decrease in pteropods biomass. So what does it mean? It means that even if the acidity is lower, so better conditions in acidity for them, the high temperature implied by the marine heat wave have reduced their survival. So in BioEcoOcean now, we are trying to answer the question, are we able to reproduce the impact of global warming and ocean acidification on the pteropods with a numerical model? So we developed a model to try to simulate the evolution of pteropods in time. And we are working on the impact of global warming, ocean acidification, and we try particularly to model this blob. To do so, we are using the model CPODIM. So CPODIM LMTL for low and mid trophic levels. Low and mid trophic levels that refers to zooplankton and micronecton, respectively. And this model is based on an energy transfer through the food chain. So from the NPP, which is the net primary production, it's basically the phytoplankton at the base of the food web. And so the energy transfer to the low and mid trophic levels, zooplankton and micronecton. We have a 1D version of this model called CPOPIM that is able to simulate zooplankton biomass based on 3 environmental variables: the temperature, the net primary production, and the euphotic depth, which is the depth in the water column where there is still light. So now we can use CPOPIM with an optimization tool that has been recently developed in the team that integrates data from zooplankton nets to adjust parameters so that the model accurately reflects the observations. So build upon this modeling framework, we adapted CPOPIM for the pteropods. So far, the model was considering all of the zooplankton species, taxa, etc. And now we are implementing new processes that are specific to pteropods, such as a temperature and acidity-based mortality and habitat index. And we are optimizing this model with the observations that we have on pteropods biomass at Papa Station that you've seen on the previous slides. So now the question is, does this model work? So here you have the first result we had. It's a comparison of the simulated pteropods biomass, so from the optimized model, compared with the observations. So in the dark blue line, you have the optimized model, and in light blue shape, you have the observations that you've already seen. So here we observe that we managed to reproduce the seasonal cycle of pteropods biomass, but we do not retrieve the magnitude of the highest peaks in the observations. Now, specifically about the blob, we observe a decrease in the modeled biomass of pteropods, which is good, but the decrease is really less significant than the observed biomass decline. This could be explained by 2 points. First, we are using a 1D model instead of a 3D model. And the Papa Station is located in an area where the comparison between 1D and 3D models have shown the greatest differences. And the second point is more of a perspective. We could try to use different phytoplankton functional types or phytoplankton sizes to force the model. As the pteropods are filter feeders, they are very impacted by the size of the available phytoplankton, which could be really impacted by a marine heat wave. So that is preliminary results. It's ongoing work, and we are going to work to improve this model in BioEcoOcean. So as a conclusion, the take-home messages of today, what is at stake? The ocean acidification is a real threat for calcifiers, so for pteropods induced. And we could have cascading effects through the food web. So if we have a decline in pteropods biomass, it's likely that we'll have a decline also in the higher trophic levels biomass. And we could also have an impact on carbon export and sequestration. So in BioEcoOcean, we will try to improve the ability of the model to faithfully reproduce the impact of global warming and ocean acidification on shelled pteropods. And so our first perspective would be, as I just mentioned, the inclusion of different phytoplankton functional types in the model. And once we'll be happy with the model, we would like to derive climate projections to be able to have more insights into the evolution of the pteropods in time under changing climates. Thanks a lot. I guess we'll go into questions now.
Yes, thank you very much, Lara. I can see that the questions and answers box is still empty. So please just go ahead and type your questions And meanwhile, I think Lina wanted to ask something about the model improvements and what could be done. Maybe you can ask the question yourself, Lina, if you would like.
Yeah, absolutely. Because I'm curious. So what is it that we need to make the models better? I mean, what do you need as a modeler to make it better?
Data. We need data. Data in different sites, data that we can compare, data at different depths in good quality. As I said, now we have an optimization tool for the model that works quite well technically. And the more data we have, the more we can train the model and the more accurate we are in our predictions.
A little related to that, I was wondering, because now for pteropods, you use temperature phytoplankton and the ocean acidification. How well is it actually known, for instance, temperature restrictions for pteropods? And is it that type of data you might need if you would like to be accurately able to predict, for instance, jellyfish or, I don't know, some harmful algae, things like this?
Yeah. So for pteropods, there are a few papers that mention the conditions needed for the habitats in temperature and aragonite saturation. So we have a curve that we just can follow to have the habitats. So it's quite known so far. But that's this kind of things also that we need to implement the new processes that are specific to each functional groups. So spodym is a global model for zooplankton. Now we implemented specific processes for pteropods, so including the aragonite impacts. And that's the kind of things that we need to know quite precisely for others functional groups. So for instance, for the jellyfish, that's one of the next steps that we're going to do in BioEcoOcean too. And so we have to understand quite well the habitats, the processes of mortality recruitment for each of the species that we want to study.
All right, thank you. Now we have some questions popping up here. Dina is asking, what is the current readiness level of biodiversity modeling? How long we still need to be able to rely on models and any limitations down the line? That is that is the question, I guess, what we all would like to be able to answer.
Yeah.
But give it a shot.
Are we talking about all models generally or about my model? No,
well, we can start with your model and then any model like spodym, for instance, which is reproducing diversity of zooplankton as one thing.
Yeah. I guess it really depends on the location and on the data set that we use for validation. We have some areas where the spodym models works really well, like when we compare with zooplankton or microplankton data, we have a very good comparison, and some other areas where it's not that good. Is it depending on the forcing of the model or on the validation data? It's always kind of difficult to know what to improve first. I'm trying to find back your question on the chat to have the other aspects of your question. I'm sorry.
Yeah, I think it disappeared already to the answer. So wait a second, I'll take it back and then we read it. What is the current readiness level of biodiversity modeling? How long we still need to be able to rely on models, any limitations down the line? So I guess the limitation is the data issue and verification, but what would be your take on when can we actually say that we know we have enough data, we know enough of the processes that now we can just run the models?
I think we will never be like, we will always be able to improve our models, but now the ones we use here are quite good, and we can rely on them. But still, be aware of the comparisons of like with the observation data. Like we need to look at each station that we want to study, have a look at the model, have a look at the observations, and it might be quite close.
All right, that sounds very optimistic. Thank you. Then we have a question in chat, which is there any accurate guide to the idea of the life stages of microscopic images of zooplankton? I guess it's not so fair to ask this from you, except for pteropods. But do you know if there's a good pteropod guide?
I don't know. I really don't know. I'm sorry. Microscopic images for zooplankton.
Yeah, I think this depends a lot on where in the globe we are. There are a lot of guides, but they are very specific for the areas. But I would actually like to still ask from André of this. I find it very fascinating how the satellite observations have progressed. If you could just tell us what do you think is the major advance? What will be the fantastic thing what can be measured through remote sensing in the future?
I think our methods are established. Our main challenges are with having field data as also for the modeling component, and also the technology. So I think it's the field data and the technology that kind of don't let us improve at maybe at the rate that we want, but with these new advances on data availability, I think we have margins to progress, especially I think the hyperspectral data. There is a big anticipation, so I think the next years we will see many results coming out. So let's see what's come from the hyperspectral part, that you can start to access better the community, things that we hadn't accessed before. And then at the level of let's say the larger organisms, such as macroalgae and things like that, I think it's also with the very high resolution imagery that you can, for example, now Copernicus also allows you to, it has a third-party mission component, so it has access to these very high resolutions where you can submit a simple proposal, relatively simple proposal that if accepted, they granted you some credits to access this very high resolution image that you hadn't before. So there is progress on this, and I think it will help a lot to map with detail this assemblage. Yeah.
Okay, thank you. There is the one final question in a chat to you. It is from Benjamin 2. And the question goes, is it possible to predict plankton blooms from satellite data, for example, jellyfish blooms?
Well, I think so. Just as zooplankton, if they leave some color, some signature in the color, I think they can. Again, it's about having. It's really difficult to locate the. Even if you know there has occurred a jellyfish bloom, once you go to the satellites, you really want to have exact space-time locations, and this is the hardest part. So in the ocean, in the coastal ocean, how are you going to have the luck of having the really long time of the bloom? But again, it's not a very let's say well-known research topic. How are you going to detect the jellyfish blooms? But I'm sure there are some papers on this. Just the difficulty of finding these blooms. It's with the color in the signal. It's the most difficult.
All right, thank you very much. And then it is time to proceed to our next slot. And there we have two speakers. If Andy, if you would please share your screen. So we have first another modeling aspect by Andy Visser. biological carbon pump and ocean carbon sequestration. And then we have a talk from Artur Palacz about the marine organic carbon atlas. And so, Andy, please go on. Screen is yours.
Andy, we see your speaker notes now, not the presentation.
So I guess you need to click on something else to get it on a presentation mode. Yes, now it looks good. Thank you. Now it is again, it was in a presentation mode for a second. Now it's on not again anymore.
Yeah. Can you see me? Can you hear me now?
I can hear you, but I can't see you. But we can see your slides, but they are not in a presentation mode at the moment. Now we see something entirely different.
Can you see that now?
Yes, now it's perfect. We can't see you still, but we can see your slides perfectly. So please just go on.
Okay.
Oh yeah,
Here
now
we
we
go.
can also
Right.
see you.
All right, it's always a little bit stressful getting these things set up. But anyway, here we go. So I want to talk to you today about the biological carbon pump and carbon sequestration in the ocean. And essentially, while there's all kinds of really interesting topics regarding that, what I want to talk about today is perhaps this poor communication that we as a scientific community have with regards to communicating across the science policy interface. And this is a serious problem that I think should be addressed. So just to go back and talk about the oceanic, about the biological carbon pump, it's sketched out here. I think quite a few people understand what it is, but just to go through the process again, essentially plant life, plankton in the surface ocean, takes carbon dioxide out of the atmosphere and converts into organic material, a fraction of which, about 20% of which, sinks out of the surface ocean and becomes remineralized in the deep ocean, contributing to a fairly large pool of carbon that resides in the deep ocean for several hundreds of years before it's carried back up to the surface ocean through the meridional overturning circulation. And basically, this process is really important it's an important component of the global carbon cycle. And of course, it's also one of these processes that's open to perturbations by climate change. So that's the process that we're looking at. To put that into context of the important reservoirs of carbon in the ocean and in the atmosphere, this is a little sketch that I put together just to indicate the relative sizes of the reservoirs of carbon that's stored in different places. The largest pool of carbon in this whole system is the total amount of dissolved inorganic carbon in the ocean is about 47,000 gigatons, which is enormous, and it's basically that huge capacity of the oceans to store carbon that makes it so attractive for understanding or perhaps mitigating anthropogenic emissions. Is by far and away the largest pool of carbon that you can sort of see here. The total amount that's in the atmosphere is much less. It's about 40 times smaller, even with today's standards. But most of that dissolved inorganic carbon is just simply kept in the ocean by physics and chemistry. It's just that water is a fantastic absorber of carbon dioxide. You just have to look at your carbonated drinks to understand that. There are other pools within that dissolved inorganic carbon pool. There's the respired carbon, which is basically the product of the biological carbon pump. So there's about 2,000 gigatons stored there, which is small compared to the total amount, but still quite large compared to the amount in the ocean, in the atmosphere. And that's what I'll be talking about today. There's also other sort of like biogenic carbon. There's dissolved organic carbon. which is basically the leftovers of life, all kinds of material, lipids, carbohydrates, all kinds of other stuff that's sort of sitting dissolved in the ocean. It's about 700 gigatons there. And then there's carbonate, the biogenic carbonate shells and other things that are in the ocean. There are marine sediments as well, but there's two other little points that I might just talk about. There are vegetated coastal ecosystems, so these are, if you will, salt marshes and kelp forests and seagrass meadows, and they store about 25 gigatons. And in comparison, there's a very small dot there, and that's the total amount of living biomass in the oceans, 5 gigatons there. So you can think about that 5 gigatons of living biomass as being responsible for maintaining this enormous great pool in the ocean of biogenic carbon. Basically, when we're talking about ocean carbon cycles and climate, what we are particularly interested in is, are any of these bubbles getting bigger? In other words, if there is an expansion of any of these bubbles in this plot, then potentially that's an offset for anthropogenic emissions. Just to give an idea of what the overview is on where these anthropogenic emissions have gone, of the 460 odd gigatons that we have released into the atmosphere since the Industrial Revolution, about 280 gigatons has gone into the atmosphere, 160 gigatons has gone into the dissolved inorganic pool in the ocean, and about 25 gigatons is also in the dissolved pool, but residing in the surface ocean. One of the really important things to see about this is that within the bounds of uncertainty, all of the carbon that has been taken up by the oceans can be attributed simply to physics and chemistry. There is little evidence, I mean, really no evidence to indicate that the biological processes in terms of either biomass or the biological carbon pump have played any role in taking any of this anthropogenic carbon. And one of the things that is one of the things I want to take up is this propensity of the scientific community to talk about processes in the ocean as if they are actually taking up carbon out of the atmosphere as an offset. The entire literature is full of claims that the biological carbon pump and other blue carbon habitats sequester carbon. And this is just a whole bunch of papers just that I've lifted from the literature. And I don't mean to embarrass anybody by this. I mean, my own papers are amongst those. So there is this idea that perhaps we are utilizing or saying something that is not particularly correct, because who do we believe if there is no actual carbon being taken up out of in any uptake of carbon emissions through the biological carbon pump in in the large scale? Yet we talk about this incessantly. So what do we believe? So one of the things is to really gauge what is understood by carbon sequestration by the rest of the world. We as marine scientists might have a very particular and nuanced view of that, but what does actually the rest of the world understand as carbon sequestration? So I asked AI, and I'm not advocating that we use AI as a research tool, but it's just a fantastic means of trying to get some general ideas. And you can sort of see here what the general idea is about what carbon sequestration means. It's means of mitigating climate change by reducing the amount of carbon dioxide in the atmosphere, capturing carbon and storing it to prevent it contributing to climate change. Capturing carbon dioxide through technological means to help reducing greenhouse gases and mitigating climate change. So in the deep analysis, carbon sequestration to the rest of the world means an offset to anthropogenic emissions. But how do we as a marine science community use this? Well, there has been this statement that's crept into the literature for the last 10, 15 years that carbon sequestration is removing carbon out of contact with the atmosphere for more than 100 years. And the way that we go about gauging this is that we look at the biological carbon pump, all this material that's sinking down, the various particles or, migrating animals. And we draw basically a line somewhere in the ocean, and then we can sort of say any carbon that goes below that stays there for more than 100 years, and above that it's less than 100 years. And so sequestration is simply the rate at which we are actually putting carbon into that deep pool where it stays more than 100 years. But there is an enormous logical fallacy here, because this does not necessarily mean that that bubble changes. Essentially, it could just as easily be replacing carbon that has been in that bubble for 100 years. In other words, there is also a chance that whatever goes down is also coming up, and there's no way of saying which is, whether that's true without going to additional information. Essentially, we've been using carbon sequestration to simply talk about the turnover rate of long-lived carbon reservoirs. And this has, become a little bit of a problem. Trying to find where that attribution comes from, carbon stored out of contact with the atmosphere, it can trace it back to the IPCC in 2007. They talk about sequestered carbon as carbon stored out of contact with the atmosphere for more than 100 years. And that is a perfectly fine way to define a reservoir, one of the bubbles in that plot. But making that an active verb and sort of like talking about carbon sequestration as that removal is not necessarily correct. It does not preclude that the carbon that's being stored is simply replacing carbon that has already been in that reservoir for 100 years and now is leaking out. It is, if you will, from a mathematical point of view, it's a necessary but not a sufficient condition. In the same set of documents that the IPCC also states that carbon sequestration is defined as a process of increasing the carbon content of a reservoir or a pool other than the atmosphere. In other words, that speaks directly to what I was saying right at the beginning, is that are any of those bubbles in the reservoir pools in the ocean getting bigger? And the way that we should address it has got to do with time scale. So if you think about any particular storage unit in the ocean, whether it's the deep carbon pool due to the biological carbon pump, or for that matter, a carbon reservoir associated with a blue carbon storage in the coastal system, there are three basically important timescales that we have to consider. There is the reservoir lifetime. How long has that reservoir actually persisted? How long has that been there? And that sort of gives you an idea about it gives the lifespan of that reservoir. Then there's the perturbation timescale. So how fast are we changing that? Is that being changed rapidly or slowly? And then finally, and most importantly, there's the residence time. Carbon going into that reservoir, how long does it stay there before leaking out again? And by looking at those three different timescales, we can categorize different reservoirs as being either novel, being new, and therefore filling up, or being in steady state, and therefore simply a process where it's flux in very much equals the flux out. So essentially, most natural carbon reservoirs that we're talking about, and that includes the reservoirs associated with biological carbon pump, they are in steady state. So whatever that flux in Q is, it's simply a turnover rate. It's got nothing to do with whether that bubble is getting bigger. In order to explore that, we actually have to look at a different metric. In other words, we have to look at whether that flux is becoming larger or smaller, or by extension, whether the residence time of carbon in that reservoir is getting longer or shorter. So that's a little bit of a strict mathematical detour. But essentially, we can also start to look at metrics that can give us a better idea about whether the oceans are actually storing extra carbon or not. And one of them is the oxygen constraint. It's right there in the equation. Basically, photosynthesis goes one way and respiration goes the other way. So for every molecule or mole of organic carbon that sinks into the deep ocean, it is respired, and it draws down a mole of oxygen, roughly speaking. And through that, we can actually observe through long-term monitoring that the net amount of oxygen in the ocean has actually gone down by about 0.1 of a gigaton per year over the last 3 decades or so. To put that in context, it's about only about 1% of our current emissions. So there's not really a huge amount that's being added to that. And tracing back, the consequence of that is probably due to increased residence time. It's not got anything to do with the biological production so much as the fact that the oceans are becoming turning over somewhat more slowly, and so what goes down stays down a little bit longer, and that's where that increase in the oxygen demand in the deep ocean comes from. There's also another thing that we can use as a test, and I call that sort of like the Holocene constraint. We're coming off the back of 10,000 years of extremely stable climate, where the temperature, and for that matter, the carbon dioxide content in the atmosphere has been extremely stable. And it's only been in the last 75 odd years where we've gone through this steep acceleration in the Anthropocene, where we've started to inject all kinds of greenhouse gases into the atmosphere, but all kinds of other things. We've also perturbed the entire system in other ways. But during the middle of the Holocene, everything was ticking over really quite nicely. And if you really want to go back and find a pristine, or think about a pristine Earth that we can aspire to in terms of conservation measures, the middle of the Holocene is probably a good model for this. So the entire system was working well. The natural cycles of carbon were working perfectly well. And what I've sketched out here in these 3 other lines, the A, B, and C, are so-called sequestration rates that have been reported in the literature. Now, had those sequestration rates been operating during the Holocene, they would have stripped the entire atmosphere clean of carbon dioxide, precipitating a snowball Earth, which clearly did not happen. So whatever else we can say, we can discard those as actual sequestration rates, and sort of like they are probably more got to do with turnover rates in terms of what goes in comes out again. Where we are right now, we are in a political. So I just want to make the point that what I'm talking about is not simply a semantics. It's actually vitally important that we get this messaging right. We're engaged in a political debate concerning climate action, where the term sequestration actually carries a lot of weight. As you can sort of see from the analysis from AI, or if you would ask anybody, you would sort of like see that sequestration is interpreted by the rest of the world as indicating it's an offset for carbon emissions. There are obviously instances where well-meaning conservancy agencies have sort of like misinterpreted scientific results and put out statements which are clearly false. This, for instance, what I'm showing here is that every year coastal wetlands, for instance, sequester enough carbon to offset the burning of over 1 billion barrels of oil. Well, that's patently false. It fails the Holocene test for a start. But basically, that's kind of the understanding that we have. And I know exactly where that comes from. It comes from a very unguarded use of the word sequestration in the scientific literature. There are also instances where scientists have actually perpetrated their own misuse of the term. And there are 2 examples here, both in very prestigious journals that have gone through the peer review process and got published anyway. And the first sort of like claims that, for instance, Australia does not have to do anything about its carbon emissions. They can simply trade on the potential of their blue carbon reservoirs on their extensive coastal areas to soak it up. And the second one argues that the validation of the biological pump by the islands of the Pacific Ocean could fuel their economic development. So with issues such as climate-based solutions in maritime and marine carbon dioxide removal high on the agenda, it's vital that we as a scientific community are actually really transparent in the way we use this language, because it seems that it's colouring not only the political debate, but it's also confusing us ourselves. So that's, thank you for your attention.
Thank you, Andy. We will jump directly into the presentation of Artur. And meanwhile, I would again like to remind you all to use the questions and answers box for your questions. We take them after Artur's presentation.
Can you see my screen and hear me all right?
Yes, everything is fine. So just go on.
Okay, so good morning, good afternoon, everyone.
Artur,
Yes.
now I only see your screen myself, basically. Is that the problem for everyone?
No, I see everything well.
Okay, good. Then Artur, please just go on.
Okay, so good morning, good afternoon. My name is Artur Palacz. I work at the Institute of Oceanology, Polish Academy of Sciences, EOPAN. And this presentation is on behalf of my many colleagues at EOPAN, at BioEcoOcean, and other collaborators. So I want to start by sharing with you an imaginary news article, which I was asked by our project coordinator, Lina, to write at the beginning of our project. And I found this a challenging but inspiring exercise, which basically forced me at the to imagine a future in 2050 with respect to my work. And my vision was to be able to obtain a four-dimensional live view into the ocean to observe and predict the flow of carbon in time and space in all of its forms, from dissolved gas and dead particles to marine life in the water and the sediments. And since then, actually, in my daily work, I keep thinking how I can add maybe a small contribution to making such a vision a reality. The idea of creating a marine organic carbon atlas has from the beginning been inspired by the enormous success of two flagship data synthesis products of the global marine biogeochemistry community, the Surface Ocean CO2 Atlas and the Global Ocean Data Analysis Project, GLODAP. And both of them started as bottom-up community initiatives among field-going scientists who wanted to deliver the highest quality, comparable data products which would meet the needs of the modeling and observing communities and those of climate assessments. And today, actually, few people studying the ocean carbon cycle can imagine a world without SOCAT and GLODAP, which on an annual basis provide state-of-the-art global maps of key biogeochemical essential ocean and essential climate variables. However, there's still very little synthesis data on the organic part of the ocean carbon cycle, so about the role of a marine life in it. We know that this is a very important knowledge gap, irrespective of whether biology plays a role in carbon sequestration or not. For example, based on the work of the Integrated Ocean Carbon Research Expert Working Group of IOC UNESCO, which in 2021 published a report which, among other things, outlined key outstanding questions for the next decade of ocean carbon research. So for the purpose of this talk, I want to highlight two of them, which are especially of relevance to the biology and ecosystems community. One is, what is the changing role of biology in the ocean carbon cycle? And what are the exchanges of carbon between the land-ocean ice continuum? And how are they evolving over time? So answering these questions requires unprecedented collaborative developments among the observing and modeling communities. Providing understanding and modeling capacity to help answer these and other questions is the key motivation behind the proposed Marine Organic Carbon Atlas, or MOCA in short. So the aim of MOCA would be to provide data-driven estimates of organic carbon stocks and fluxes, both living and dead, across the marine domains from surface to bottom, from coastal to open ocean. Now, I know this sounds like a mammoth task, so to make this more realistic, we have in BioEcoOcean started to develop a pilot demonstration specifically for the Arctic, where on one hand we observe strong and rapid changes in the marine environment, and where on the other hand the challenge is perhaps the greatest because because it's so difficult to observe in the field and from space. So the diagram in the middle here, which comes from our partner project Sequester, illustrates very well the complexity of the processes that need to be considered with respect to organic carbon cycling in the Arctic or the polar regions in general. And with the smileys on the left-hand side, I indicate that while we have a much greater availability of data and data products, as well as predictive capacity related to surface ocean carbon uptake and export, much less is when it comes to water column and sediment recycling and burial. With that in mind, the conceptual development of Arctic MOCA started actually by bringing representatives of the observing and modeling communities to jointly co-design what we call the top 10 biogenic data products to advance ocean carbon sequestration modeling in the Arctic. So mind you, that workshop was before Andy's talk, but nevertheless, I think same priorities for the products apply for ocean carbon cycle modeling in general. This initiative was started in 2023 by 2 EU projects, Ecotip and EU for Ocean Ops, which through a combination of an online survey and a community workshop engaged over 30 marine observationalists and modelers from across the biogeochemistry, biology, and ecosystems domains, focusing on Arctic research. So this activity tasked us to agree on priority data products, their required scope and format, while taking into consideration both their feasibility to obtain based on available data, but very importantly, their likely impact on advancing our modeling capacity. Now, the outcome of that working group was a list of data products, which you can see here in gray boxes on this diagram. And they help collectively describe key processes and variables, many of which coincide with the subvariables or derived products of essential ocean variables or essential biodiversity variables. Now, this diagram here also maps out how these priority biogenic data products help constrain 2 important metrics describing the efficiency of the biological carbon pump, which Andy just talked about. The first is the E ratio, which is the measured or modeled ratio of net primary production to particulate organic carbon flux at a given depth. And the second one is the so-called B parameter from the Martin curve, which describes the fixed rate of particle degradation in the water column. So actually what we need is to be able to estimate in the models the variability in B due to the various remineralization and sinking processes as a function of properties, and then also compare with any E ratio estimates. As a direct follow-up from that initial activity, we set out in BioEcoOcean to design a detailed roadmap for delivering new or, in fact, sustaining any existing priority data products specifically related to the role of benthic life on ocean carbon cycling. So just last month, we successfully held another community online workshop, which brought together a group of international experts in observing and modeling, but this time focusing on benthic carbon processes. So this initiative was co-organized by actually 4 different EU-funded projects and was being led by my colleague Marta Szczepanek from EOPAN. So stay tuned for future publications and new data product developments, which will be based on the recommendations from this workshop. Now, moving up the water column, MOCA is keen on adding to the existing wealth of initiatives focused on plankton and carbon cycling. So here on this slide, I really want to highlight a recently published article by Maria Grigoratu et al., to which BioEcoOcean also contributed, and which provides, in my view, a fantastic and very broad summary of why we all as humans should care about plankton, not just in relation to carbon cycling. So I use this presentation here as a little advert, and even for all of you plankton lovers out there, I'm pretty sure you will find some new and exciting facts in that paper, so be sure to check it out if you haven't yet. So now for the next couple of slides, I want to give you a few examples of where and how we've been contributing to specific new data products related to plankton and carbon cycling. So first, my colleague Alexandra has developed a new satellite-based model of net primary production in the Greenland Sea, which is one of the most productive regions in the Arctic. Net production refers here to the total amount of organic matter produced by photosynthetic organisms, phytoplankton, minus the amount they would consume for respiration. So Alexander's algorithm demonstrated that we can provide basin scale estimates of production with decreased uncertainty relative to commonly used global products. And this is, important implications for how we actually link the changes in ocean Arctic productivity with other ecosystem and environmental changes observed there. The code of this model is already freely available from GitHub. You can find the links to that in Alexander's paper. But we are also now working towards making the actual data product and its outcomes operational, and so available to all interested users. Alexander is also leading some pioneer work on estimating net community production in the Arctic. And this, in contrast to net primary production, is the overall rate at which organic matter is accumulated by the entire marine ecosystem, taking into account both photosynthesis and respiration of all organisms and not just producers. So in this study, we use years of near continuous measurements of nitrate, a biogeochemical EOV, in the water column from a growing fleet of Argo floats equipped with biogeochemical and biological sensors. And we used those results to estimate the annual net community production in the Arctic over a number of years, actually providing first such estimates in this part of the world, not counting some point ship-based estimates from the past. MOCA also includes new field and satellite capacity to observe changes in phytoplankton functional types in the Arctic. Led by Kasia Dragańska-Deja and in collaboration again with several other EU as well as ESA projects, our team synthesizes historical data on phytoplankton pigments measured over the past decades to create better basin scale estimates of such changes based on the field, and then also now actively working towards developing a dedicated regional satellite algorithm for PFT detection from space. Taxonomic analysis of plankton remains a critical and strategic focus for our work at EOPAN and in BioEcoOcean. So my colleague Anya is thus analyzing phytoplankton species diversity collocated with pigment and biooptical observations to provide indispensable knowledge of who is actually doing what in the water, and thus providing very important ground truthing for all satellite and other model developments in this data pool region. When it comes to the role of plankton in carbon cycling, the challenge is, of course, to say, as accurately as possible what the carbon content of the different species assemblages is. And that is also a part of our long-term plans for MOCA. You have already heard from Sarah about zooplankton modeling today, but here I just want to emphasize again that our lack of sufficient constraint on observing and modeling zooplankton processes is the largest source of uncertainty for marine carbon cycling and nerve system models. So those which are used as a basis for IPCC assessments. And while our colleagues at DTU and Mercator do bulk of the work on advancing those topics, here I just want to highlight one contribution to MOCA by my colleague Lena, who has been conducting a systematic review of model representation of the role of zooplankton in carbon sequestration. And the review has identified over 80 global models, 15 Pan-Arctic models, and over 70 other regional or local models, which can be used to estimate these processes to various degrees. And such a review would provide an important baseline for assessing not just the current capacity and the gaps, but also what are the needs for further advancements in this realm. Here on this slide, I want to communicate that we're not building MOCA as a one-time off atlas. Rather, we aim for this development to be integrated into the state-of-the-art global federated and interoperable data management system. So by this token, the vision is that for you, using existing digital infrastructure, we would envision all elements of MOCA to be findable and accessible via basically a worldwide web-like search, which would be provided by the Ocean Data Information System or Ocean Info Hub, some snapshots from which you can see here on the screen. This is already an existing solution that we want the MOCA concept to be fully integrated with. So in that sense, if you're wondering how you could contribute to any MoCA developments, it is precisely by making sure that your data or model outputs are part of such a system. Now, what would that require? You've already heard a lot today about FAIR principles, but I would argue that every scientist should be familiar with what FAIR data means: findable, accessible, interoperable, and reusable. If you're an observer, you should share your data and, very importantly, metadata in a standardized format for a trusted repository. And ideally, that should be done in a timely manner so that we don't wait 10 years for some data to be used. And if possible, think about joining a community or a network that is already working towards coordinated observations and data product developments. On a global space, this is the Global Ocean Observing System, which you will hear more about also in a few minutes. But locally in the Arctic, we've got a network, a pan-Arctic network of so-called distributed biological observatories, which work towards those goals precisely. So, no doubt there's a long way before something like MoCA can become part of this all ocean observing value chain that maximizes the value of each ocean observation and actually delivers that all the way to scientific assessments. And so that MoCA can, be on the same level as SOCAT and GLODAP, which I mentioned earlier. So it is perhaps more of a 2050 vision, at least for the global ocean. But then again, as Rudy Dornbusch said, things take longer to happen than you think they will. And then they happen much faster than you thought they could. So I at least remain optimistic and invite you to be as well. Thanks.
Thank you very much, Artur. We are running a little late, but we do have time for a question or two. The question box is empty, but we have something in the chat. And we take a question from Said. And what he is asking is about methane seeps. So I read up. Hi, André. I recently came to know that methane seeps are also important sources of carbon in marine lacustrine and geologically active environments. Are there inputs considered in these carbon sequestration models?
Yeah, so carbon seeps are definitely sort of like a process that's being monitored. It doesn't come into these sorts of calculations, insofar as that there are carbon seeps that are releasing carbon or methane into the oceans right now, but they tend to be fairly long-lived, fairly slow, and there's also balanced against the input of methane. So material that's sort of like going down and being buried in deep carbon sediments. So there's a bit of a balance there. So exactly what a trade-off is about, extra carbon coming into the system or leaving the system in terms of methane seeps is still something that's not particularly well known.
All right, thank you. I think we leave the other questions in chat for the period after the next slot, not to run late. So, Lina, if you could share your screen. We have one more round of talks to go, and this will be with Lina and Anna. And yeah, maybe we just go on with it. Please, Lina.
Yeah, thank you. And thank to all the speakers already for a lot of interesting talks. For this one, it will be a little bit interactive. So I would like you to actually bring out your phones so you can scan the QR code. But I will talk a little bit, or the topic of the talk is beyond the usual suspects. And can additional variables advance your research and monitoring? So if you are bringing out your phones, I would like to hear a little bit from you about what you would measure, monitor to understand your marine ecosystem. And this will become a word cloud, and here you can provide multiple responses if you want. And hopefully someone wants to engage. At least we have one participant engaging. But these things are quite important to think about, right? What would you measure to understand your marine ecosystem? Okay, let's look, and then you can just continue writing. So we see here, for example, temperature, we see salinity, depth, species abundance, species richness, pollution, the water depth. And I think this is quite interesting. A lot of these things or variables that you would like to measure here, or you put forward here, are sort of belonging to the natural environment. And because of this, I want to take you through an example. And the weather here in Sweden today isn't very good, so I'm actually going to take you to a tropical environment. So thank you very much, everyone, for submitting some things to measure and monitor. So taking you to this marine ecosystem, this case is really bringing you from the Arctic that we just heard about to a case that I would like to give about in Tanzania. And I will use a framework to help us guide us through this system and also hopefully support us in thinking a little bit more holistically about our system. And the system can be defined as whatever is important for you or in your context. It can be the whole globe, or it can be a small case study, or it can be, for example, the whole Baltic Sea. But in this case, I will take you to a bay in Tanzania. So I said I will use a framework to sort of guide us through this process. And frameworks are ways to sort of help us better or guiding our efforts. And this can be used and to better understand our system. And these can be sort of linear processes or circular processes or more complex sort of approaches. And I will sort of base this in the social ecological systems framework because it sort of really brings together the social side and the ecological side and the interactions and outcomes when we are using a system. And it really supports this conceptualizing these coupled systems, and it can really help us sort of to diagnose the system and understand it better. And I think most importantly, maybe that it can help us with communicating across different fields. So we get a sort of a shared vocabulary and we can look at this together coming from different perspectives. So this is a very beautiful woman in Zanzibar, a tropical island, and she is a gleaner. So she's collecting invertebrates here in the seagrass system. And this is really showing an interaction between a person or humans and our natural environment. And looking into the social ecological system to try to better understand this, we have this invertebrate gleaner, and she would be then the resource user in this framework. And the things that she's collecting, those are the resource units in our example here, and she's collecting them in this resource system. And the governance system would be, for example, if there are some regulations to the gleaning that maybe you can only catch this much, or you're allowed to catch or fish here, but not over there. And the social, economic, and political setting would be sort of how that community looks like. Maybe there is a very few jobs available. Maybe there is a high level of poverty. And while this related ecosystems that you see there at the bottom refers to the natural environment that is sort of surrounding your system. And this could be then, for example, wind speed and the mangrove swamp. So I want to walk you through sort of this example here. So by using this social ecological system, it can really support us to think more holistically about our system and what's affecting it and the interactions and what it will lead to, sort of the outcomes. So here we have our resource system in this example. And this resource system is, of course, not in a silo. It is surrounded by our natural environment, the so-called the related ecosystems, and also the physical things that works in these systems, like, for example, rainfall. And in our resource system, we have these resource units. It's what we have decided is important for our sort of analysis. And in this case, these are the species that are being targeted for this type of fishing. And we are, of course, in a social, economic, and political setting. In this case, it could be sort of access to market, or availabilities to public transport, etc. And then also, of course, the governance system, the rules and regulations, the different approaches to steer human behavior in certain ways. And most importantly, that's maybe the wrong word, but most interestingly, it's all of these interactions between all of these different components in our system, and what these interactions, what kind of outcomes they lead to. So, for example, I think we're all aware that if we are overfishing, we put a lot of fishing pressure, it might reduce the size of our resource units, in this case, the bivalves, and it might also affect our resource system in a way, sort of losing, in this case, seagrass. And hopefully this sort of sparks the governance system to maybe put in place that we need to reduce the fishing pressure. At least that's in an ideal world. So I think it's sort of the purpose here is we have both a sort of a natural side to this and a social side. And it's these interactions between those that can really sort of affect our environment and as well as our social system. And you had that first slide typing in what to measure, what to monitor. And what we often see is that the focus is very often on this ecosystem side, sort of the natural system side, that we are talking about salinity, we're talking about maybe the number of resource units that we have, or the distribution of our habitat. And we are seldom including sort of the social variables, the social drivers that might be extremely strong drivers for change in your system. So I think this is something I really would like to encourage us to have a more of an open mind, that we also need to really put a stronger focus on looking into sort of the social side of the system, and also monitor and measure those variables, because they are also very much affecting our marine environment. And I want to also highlight a paper that was just published 3-4 weeks ago, led by Niklas Hode here, one of my colleagues, about taking advantage of open data in coastal science and conservation. And And in this paper, we have also used this approach with the social ecological system to help us, to guide us, to find variables that could be relevant for a system. So we started by developing a workflow, talking about sort of identifying the site of interest, sort of your system, and then using these social ecological systems framework to identify potential variables of interest in your context, and then look into open data or what's available as open data, and if that can be used to sort of enrich our work. And then there is a wealth of open data available out there. So it's not always about us just publishing. There is also a lot of benefits that we can have by using the open data that is available. And then going through this workflow, this can either be used to sort of plan your field work or plan your in situ or in field measurements to either complement or maybe verify data that is available as open data. Or it could be used sort of to enrich. You have done, you collected data, and there's additional variables you could add sort of to your analysis. Or you can just use it as a desktop study or as a standalone and start looking at what's available out there. Here's just a brainstorm of potential variables or proxy variables that could be very useful, in this case for the seagrass system that I just showed you. And we know, for example, for seagrass, it says there up to your right corner, terrestrial protected areas. That might sound strange, but we know for seagrass that protection on land can have a very strong effect, positive effect on seagrass, for example. So this might be a very important variable to include. Here's a more organized way of sort of structuring this, and this figure comes from the paper, to really start mapping out which variables, or in this framework they're called indicators, but they're the same thing. And these indicators or variables, they can also be mapped out. And here is an example, for example, mapping distance to port, which could be very important if you are fishing, for example. It can also be used as a sort of a proxy for fishing effort. Different types of water surface or surface temperatures. There is available data on max, min data, etc. Subnational Human Development Index can be a very strong driver for what's happening in your marine environment, etc. And before we end, I just want to put up another Slido, and because I would really like to encourage you to think of any additional variables that you think could be beneficial to measure to better understand sort of your marine ecosystem that you are interested in. And if you have some examples maybe of things that you think, thinking a little bit more holistically about the system that you could add. And this one comes up as text boxes. Now I'm advancing here, so. And we can see, for example, distance of nearest human infrastructures to the coast. I think that is a very important variable to look into. And also something that I would like to say is that even if your system or your area of interest is in the middle of the open ocean, humans have, or we, the sort of the social system, have a very strong impact as well. And I think the further we go from the coast, we use less and less maybe social variables or social input, trying to understand our systems. So this is something that I would really like to encourage you to do, to think a little bit more broadly about your system and what to actually include, or which variables to include to try to better understand your system. So thank you so much, everyone, and also to everyone that has responded to the Slido. Thank you.
Thank you, Lina. And we go straight to the next presentation and the last presentation of the day by Anna. Thank you very much. It's early times, I guess, or late, one or the other.
Thank you. Thanks, Marja. Can you see my screen, the presentation? Yeah?
Yes.
Perfect. Hi, everyone. Good morning, afternoon, good night, evening. I'm Ana Lara Lopez. I am the International Project Officer for the GOOS Biology and Ecosystems panel. So, so we had a series of talks, very, very interesting, on some of the important work that is happening in the project BioEcoOcean. So let's try to connect the dots in here. So that's what I'm going to do, an attempt of sort of trying to provide an overview of how this work that is happening at regional level has influence of what we can achieve at a global level. So, okay. First off, we have big challenges. So biodiversity loss is one of the triple planetary crises that have been identified by the United Nations. And we are living it now. We can actually see by all the news that you can see in there that I put that we are reaching tipping points. We are looking at how the ocean is changing in real time. And with that decline of biodiversity, we'll have the potential of losing some of the ecosystem services, which are very important for our well-being, including, you know, the oxygen production, including the food production. There's over 3.3 billion people that actually depend on the ocean for food. So we need to address that. Over 70% of our planet is ocean. So it is vast. It is a very big ecosystem, and not one country can effectively observe and collect information on its own. Data and observations are very important for us to understand how we can actually address these challenges, understand what is happening to our ocean, and see how we can actually restore or reverse some of the changes that are happening. So ocean health is really important. A healthy ocean, it is key to our well-being and survival. So observations take place at a national level. So it is the different nations that are actually, due to diversity of needs, trigger those observations or those monitoring programs. So it could be for regulatory reasons, it could be for management of resources, or, you know, environmental requirements or compliance. However, we have been taking, and we've been observing the ocean for quite some time, for several years, but we have a fragmented approach on how we collect observations. We also have very diverse metadata and data standards. We have poor access to biodiversity data, and we have heard from some of our colleagues that are developing these modelings, what will help develop these models and improve their skill, data. So poor access to ocean biodiversity data is an issue. And we also have significant gaps in observation. So that makes it really hard and difficult to have data that is comparable from one region to the other. We have insufficient information to assess the health of our ocean at different scales. And we have also inequalities when it comes to accessing the monitoring capacity, the capability, and information. So, and no consistent vision of the information and services needed for users. So there's where GOOS is trying to help in coordinating how we collect observations in a standardized way and promote the fair and open data access, so we can then develop those products and we can provide the information for the end users, so they can take informed decisions based on good data. So we can have data that is comparable across regions, that it can be taken once, one observation taken once, used multiple times for diverse purposes. And so in here you can see what we call a value chain. So we're starting with the observations, moving into data products, and then the uses. So in the observing and collecting observations, you have heard through some of the presentations about the use of the essential ocean variable framework. So that's the framework that GOOS developed to try to coordinate the communities that collect ocean observations, so we can have key measurements that will help us understand our ocean better, but collect them into a harmonized way, so we can actually have comparable data. So that's the essential ocean variable framework, and you can see in there it's multidisciplinary, and alluding to what Lina is saying about the human dimension on that. There's maybe a couple of the essential ocean variables that touch a little bit on that, which is the marine debris and ocean sound. But in here, I'm going to be focusing more on the biology and ecosystem essential ocean variable, which is the one that we're working on with BioEcoOcean. So what is an essential ocean variable is the minimum set of ocean variables or ocean measurements for biology and ecosystem identified to help understand and forecast marine life. It provides that framework to coordinate how we collect the observations, so we can have globally comparable data. So we can upscale the observations that we collect and join it with all other observations from other regions, so we can do a global assessment. And I think Florian gave a really good example with the jellyfish work. So, and why do we want that? Well, one, to start understanding how the health of our ocean, but also it can help as well our nations meet the targets from different international agreements, such as the 30 by 30 or the Global Biodiversity Framework from the CBD, or the new agreement on biodiversity beyond national jurisdiction, and see if we can meet those targets, and trying to meet those targets, because we really need to address the decline of biodiversity. So, and the work that you have seen with BioEcoOcean in developing this blueprint is really helpful, because it is, while it is something that it's a project in being developed in Europe, the system, as it gets refined, is something that we can actually, it's a tool that we can apply then globally. So it goes from a regional or local into a global by using the essential ocean variables, helping us update the specification sheets, which the EOV specification sheets provide the guidance on how you collect those observations. So BioEcoOcean is testing that for us, and it's helping us refine the information that we are developing to provide at a global level. So the footprint in reality is global of what this project is doing. So when it comes to data, fair data, open data is what we actually focus and encourage. And we use OBIS and ODIS as the system to handle our data and metadata. And with ODIS, we have the BioEco portal, and in that portal, it's a window to biology and ecosystem monitoring programs. So monitoring programs, observing programs that are 5 years or older and that are collecting information on the EOV. And that allows us to see who's collecting what where, and allows us to identify where the gaps exist. And so if any of you know of programs, monitoring programs that are not in the BioEco portal, please let us know. So through BioEcoOcean, we have refined the information and the guidance we provide by developing this new section on data, on how do you submit your data and metadata to the portal or ODIS? How do you, what is the standard for submitting your information, your data into OBIS? We use the Darwin Core, and it's also helping us develop tools, different tools that will actually facilitate everyone from anywhere in the world to enter their metadata into the portal so they can be visible, and to submit data into OBIS. And that not only is if you want to submit directly to OBIS, there is also some information on how you connect your system. So if you use your National Ocean Data Center to submit your data, how can we help to connect that data center into the OBIS system so it can be visible, so we can then have the data available, and we can use it and compare with other regions as well, so we can have this global assessment. And you heard, like, having the data available then allows us to develop the different models and products that are going to digest the data and give that information in an accessible way to the users, to decision makers, policy makers, or industry, etc., whoever is the user of that data. So, and we have seen that through some of the talks that we have from BioEcoOcean. The living labs are working really hard on looking at the modeling, improving the model skill, indicator development, the right products as well that are going to be super useful for them provide information that it's fit for purpose to the user needs, co-design approaches. So there is a lot of activity happening in this project that are going to help us a lot in how we actually provide the guidance and help coordinate the observations in a way that actually can help us understand the health of our ocean and help us use that information for the multiple purposes that we have. And that will be through the early warning systems, operational services, marine spatial planning, resource management, fisheries management, etc. And among those developments as well is for the first time, we are actually having near real-time biological data on some marine mammals. So this is, I'm going to finish in here where it is really important to understand that whatever effort you can do locally, it's going to, in getting this standardized collection of observations of fair data and open data, is going to have a global impact. So it is important that, you know, when you're out there in the field collecting your observations, we have a framework to help you. And we're there to help you. You can use the Essential Ocean Variable Framework. You can use the specification sheets that we are actually developing, updating at the moment to guide you through your work. And with step-by-step information on how you can submit data into EOVs and metadata into the portal. And this is really important information that it's going to help us at a global level and also help us understand how we can actually address the challenges that we have now and that we can see at the moment developing in real time. So I will finish with that. And yeah, any questions?
Thank you very much, Anna. We have no questions so far in the questions and answers box. And I would just like to encourage everyone that you can also now post questions to all the panelists into that box. So please just do this. And meanwhile, I actually have one question for Lina related to this socio-economic variables. Could you give some examples of how these could be monitored if we are talking about things like which are related to human behavior, for instance, picking up shellfish or artisanal fisheries, even things like this?
Yeah, I mean, for example, you could keep track of where the fishing fleet is going, and sort of their fishing effort. To also sort of monitoring any changes in sort of legal frameworks or rules and regulations. For example, sometimes we have temporal areas that are closed for fishing, or areas where we might have exploitation of some kind, maybe trying to find stuff in the sea bottom that we might not want to see. The BB&J will be also a very interesting thing to keep track of. There's also a lot of different sort of money sources maybe going into different initiatives in the ocean that you want maybe to track or monitor and see what they're doing.
All right, thank you. The Q&A box is still empty. So maybe then I would like to ask all the panelists to put their cameras on, and we can take the few final questions for the full panel.
Yeah.
Yes, Lina.
So I just want to thank Anna for this good sort of tying the bag together here at the end. And I think it cannot be sort of highlighted enough that me bringing or publishing my local data, it's not just for everybody else. There's also a lot of gain, because if the other people in that local area is also doing that, they might have measured, I mean, I love seagrass. They might have measured the seagrass in April, and I'm doing that in August. And because this data is available, both of us have more data available to trying to understand our system. So it's really a non-selfish, but still a lot of win-win to publish your data. Because if we want to really make a change for the ocean, we really have to make this change and try to make more data available, so we can have all of these better models, that Sarah was talking about, for example, and Artur as well, that we need this data to understand better, as well as André. Thank you.
Thank you. That is also a very satisfactory point of it, that your data can be used more than once, right? So that is, yeah. Thank you, Anna, for the talk, and thank you, Lina, for the question or for the comment. I have a few, one particularly question in the chat for the modelers, and that goes like this. Often the observations are not at the resolution needed for the models. Is there an ideal resolution that could help improve models? I guess both in space and in time, depending. And maybe if we could get a comment on that from maybe Sarah first and Andy then. And then Artur, Florian, André, Lina, depending how much you feel like modelers or observationalists.
Yeah, I can say a word about that. I would say the thinner the resolution, the better. But any resolution is already good to work with. If we have like one zooplankton net in one location, then we'll study like the area around, and it's already very valuable data. But still, the thinner the better. And also in terms of depth, that's a dimension that is not often studied and that we consider a lot for ecosystem modeling.
Andy, would you like to add something to it? You are muted, so please unmute.
There we go, yeah. So I would just basically reiterate that. I think, high resolution is better, but we need to be a little bit, how would you say, sensitive to the fact that models can only use so much data. The higher the resolution is better, but in some sense, we don't want to sample the entire Earth and have an enormous amount of data. In some sense, we want to try to be able to distill these observations into models and sort of try to make sure that we get the main processes in these models right. So it always becomes this problem, a bit of a trade-off. So yeah.
All right, thank you. Anyone else who would like to add something to this? No? All right. And we don't have any questions and answers. So then
I think.
Oh,
Artur
yes.
wants to.
All right, Anna, please.
Artur, it will be.
Oh, yeah, just
Artur, yeah.
a short comment, if I can. I think it would be really nice if, when you're planning your sampling, you actually think about inviting a modeler that you know who would be interested in using the data to discuss what such a resolution could be. I mean, this has to be flexible enough to match a specific application, but I think fundamentally we just don't have that connection in place, and we don't have that culture of co-designing the sampling schemes with the modelers in mind from the get-go. So that would be just my one comment.
And I can see André and then Anna.
Yeah, sometimes it's also not also only the resolution, but also the type of observation. For example, when you sample seagrass or macroalgae, if you don't record also the others that are next to it, for example, or non-occurrences, they are also important. So it's also on how you observe. And I mean, in the ocean now, also thinking on the remote sensing, many cases the discrete sampling is difficult for us. So maybe sometimes we prefer to have discrete samplings to tune other types of observations, such as transects with sonars or other fluorometry to get you more data along a given line. So that's my comment on this.
Thank you. And Anna, would you like to say something to the end here?
Well, with the new technologies, I think we're sort of opening some potential higher resolution, I guess, when it comes to, imaging or combination, I guess, because every technique has its bias. But I think Arthur hit it on the nail. I think it is important for people collecting the observations and modelers to be in the same room and talk to each other, because it's in there where understanding will happen on what are the needs and what can it be met by the observations, but because there are constraints as well on the observations that we collect. But importantly, I think it is just because every time I talk to modelers, it's like, we just need data. Tell me, where is the data? And that's where the open standardized data becomes important, and not only gives you the advantage of having other regions joining and having a look at the bigger picture, but you can integrate with other groups, like other biological groups, other type of information, just like we integrate with satellite data for some in some instances, and it is because it is available. So the options and the sky's the limit. What you can do.
Thank you. Lina?
Yeah, and I would like to throw in some maybe policymakers and managers into this mix, because are we making the right products? Are we developing also maybe the right models for better decision making? So it easily comes quite complex, but as you have already mentioned, the people doing the observations and the modelers, they need to talk. But we also, I think, every once in a while need to talk to those that needs and can act on this information, because they might also not know what is possible. As Anna said, the sky is the limit, or the ocean bottom, that is a hard limit. But yeah, that what do they need? Sort of what kind of model do they need to make that better decision, and what are the available options? And when we talk to each other, we might come up with new things that can advance sort of our protection of the oceans, for example. Thanks.
Thank you. So that's a call for the data users to also include themselves into the discussion between the observators and the modelers. I have here to the test. Okay, Andy, go for it.
Yeah, so I was just going to mention that what is really useful for policymakers is to be able to do what-if experiments in a model. It's not simply sort of like saying what's going to happen in the future, but they want to have a means of testing policy, right? So if there's some sort of process or some sort of intervention that they have that they could use, they want to be able to use that and sort of like make projection to sort of like have a trade-off to see what the outcomes are. And I think that's really where we really need to have to put some more effort.
Thank you, and that is also something what BioEcoOcean is contributing to. And any more hands up? Then it is now, or questions. I have one final question to Lina, which has to do with BioEcoOcean, because we've talked today, we have heard talks from observations to data, products to models. And a big part of BioEcoOcean purpose is to develop or contribute to the best practices and the way how this can be done in a most optimal way, so that it will serve the policymakers, but it will also serve the scientific community and so on. And Lina, I was thinking if you could here in the end maybe highlight a few things what BioEcoOcean is doing to promote those best practices.
So let's start with the blueprint. So that the question-based guide that we are, or support tool that we are developing, it will spark sort of the the conversation around, should we use best practices? Are there best practices available? Being able to point to such resources. We're also developing, or during this first 1.5 year of the project, we're also testing a lot of different methods, sort of field testing them to see if they work, and being able to contribute to these best practices. When it comes to the EOV specification sheets, they have now been updated under the leadership of Anna here, and we also really need to test this, right? And then see what is available. I mean, there's a whole repository available, the Ocean Best Practices System, where you can find a lot of these different types of best practices. Yeah, I think I'll stop there. Maybe Anna has something to add. I do want to mention that, or bring up what Anna said, that we are also developing a lot of learning resources. For example, this metadata app. Instead of having to submit your metadata through a really difficult and technical approach, you can do that by just filling in a form online, and it will create this metadata file for you. So we're also very interested in sort of developing these tools that can make it really easy for people to follow FAIR principles. We're also at the moment developing for seagrass sort of a really step-by-step video on how to submit your data to OBIS.
Thank you. And I think the listeners can follow up on BioEcoOcean webpage and the social media on how these things progress. And I think then it's time to close the seminar. I would just like to mention that the recording will be available in the BioEcoOcean YouTube channel in a few days. Also, everyone who would like to see that. And then it's just left to thank you for all the speakers and all the people asking questions and all the listeners in general. Thank you for participating, and I hope you have enjoyed it as much as I did. And have a nice day. Thank you.
Thank you. Bye, everyone.
Thank you. Bye.
Thank you. Bye-bye.